Alright, we're now on to our next section on the urinary system where we were going to pick up and start talking about nephrons, which are the functional units of the kidney. And as with many of the other structures we've seen as well, there are going to be many nephrons per kidney. And they're going to be fairly consistent among all vertebrates. Now, when you start looking at vertebrates that are a bit more, uh, I guess, somewhat dependent on being around water more, so they're maybe a little bit less inclined to dry out, especially fishes, and with things like amphibians especially. They're not going to have as extensive of a network of nephrons as what, say, mammals and birds do. Mammals and birds, for example, have this really long and well-developed what's called loop of Henle, which dips down from the cortex into the medulla of the kidney. Those nephrons are going to allow mammals and birds to, especially mammals, to super concentrate their urine and really allow them to get rid of all the waste we talked about, do all the regulation we talked about back in the previous section, with a minimal amount of water. So it's going to allow them to get rid of maximum waste with minimum water expenditure, which is of course a good thing if you're living in kind of a dry environment. When we look at vertebrates producing urine, the urine is going to be either iso or hypotonic as compared to the blood. So the blood would either be isotonic or hypertonic as compared to the urine. So this is for all vertebrates now. And then birds and mammals now, like I mentioned a second ago, are the only ones that can make their urine hypertonic as compared to the blood. So when I mentioned before, they can super concentrate their urine. What effectively that means is they're making their urine very hypertonic. It's more concentrated in terms of solute than the blood is. There's more excess ions, there's more excess H pluses, there's more excess amino acids or glucose. Hopefully not, but there could be, especially in a diabetic patient. There are more of those solutes in the urine than there is in the blood, and that actually takes a fair amount of work and a fair amount of energy expenditure to make that even possible. And as I mentioned before, some vertebrates like amphibians and reptiles and so forth can't do it at all. So we can make our urine relatively hypertonic as compared to the blood fluid or the body fluid, and what that means, or the consequence of this, comes from the fact that we have a loop of Henle, which is that long tubule that dips down into the deeper part of the kidney and allows for a further concentration of the urine beyond what other vertebrates maybe can do. Birds aren't quite as good at mammals at doing this. They do have a few, typically nephrons, that are good at super concentrating urine or making it more hypertonic as compared to body fluid. But they also have other ways of disposing of, say, excess salt. If you look at a seabird like a gull or a tern or something like that, birds are exposed to large concentrations of salt all day, every day. They can have, for example, salt glands up around the eyes. You see the eye socket or the orbit here. There's a salt gland that helps to pump excess salt down through this structure here and eventually out by the nares or by the nostrils of the bird's beak. Again, since they can't make their urine as concentrated as what mammals can, this is the next best thing. They still make their urine somewhat concentrated, and then whatever other excess salt they might have in their body, they can dispose of using their salt glands. So if we look at the overall kind of macroscopic view of the kidney, we can look at the adrenal cortex, which is the part of the kidney which is kind of out here. It's a lighter shade of kind of a pink color out here. This is the renal cortex. There is a renal medulla, which is the deeper part inside of here, which has got kind of a deeper coloration in this picture. And then a structure called the renal pelvis, which is what right here effectively drains the kidneys. And from there, once we get into the renal pelvis, this is like a funnel that gets the kidney filtrate or urine now into the ureter, which then carries the urine out to the bladder, and then from there to the urethra, as we talked about in the previous section. So those macroscopic views. If we take a really close zoomed in view of this, if we take a look right here, for example, we could see cortex, we see medulla. Over here we see cortex, we see medulla, and we see the actual structures of the nephron itself. So everything from the glomerulus to proximal and distal arms and collecting ducts as well. So the nephrons are the functional unit of filtration of the kidneys. There are several structures here. There's a structure called the renal corpuscle, which is made up of the glomerulus and Bowman's capsule. We talked about the glomerulus in the previous section. Bowman's capsule is also adjacent to the glomerulus. We also see the renal tubule, and tubule is responsible for the most part when it pertains to reabsorption and secretion or getting rid of waste or ions and those sorts of things. That's going to be involved with the renal tubule. And the longer that tubule is, the more filtration and the more reabsorption that animal can do. So we look at the proximal convoluted tubule, which is the section closest to the renal corpuscle and the glomerulus especially. We can also look at the loop of the loop or loop of Henle or loop of nephron, which dips down from the cortex into the medulla. And then we can look at the distal convoluted tubule, which is further away from the glomerulus, and then eventually from there it's onto the collecting duct, and from there into the ureter. Okay, so that's kind of the overall structure or pathway of movement of filtrate, starting off at the filtration step with the renal corpuscle, and eventually moving to reabsorption and secretion with the rest of the tubule part of the kidney. So again, if we take an even more zoomed in view, this is the one we saw previously. We saw glomerulus, proximal convoluted tubule, distal convoluted tubule, collecting duct, and so on. We can zoom in on just one of these now and take a look again at the glomerulus surrounded by the Bowman's capsule. We see proximal convoluted tubules, which are closer to the glomerulus up here. So this tube kind of wraps back and around, and eventually right here it's labeled proximal convoluted tubule. We then go down into this loop of Henle, or in this picture it's labeled loop of nephron, back up again to what's called the ascending loop. Right, the part that comes up out of the medulla toward the cortex is the ascending loop. The part back here was the descending loop of Henle. This is the ascending loop of Henle. We then get into what's called the distal convoluted tubule, and eventually to the collecting ducts. This would eventually flare out into the renal pelvis, and from there the filtrate would be carried off into the ureters onward toward the bladder. This is a more simplified view with everything labeled: glomerulus, proximal tubule, descending loop of Henle, ascending loop of Henle, distal convoluted tubule, and then eventually collecting duct. All right, so blood coming in the renal artery. Anytime you see the term renal, that refers to kidneys. So the renal artery is an artery that carries blood away from the heart toward the kidneys. Eventually what's going to happen is that urine is going to be produced from the blood. As we talked about yesterday, we talked about how certain kinds of things, if there's too much of them, can be filtered out of the blood. And if they can be used, if they're not in need right now, they could either be stored or they could be disposed of in this case. And so the urine would then go from the ureters to the urinary bladder and then onwards out through the urethra. So that was, again, in the previous section, a little bit of information we saw before that I just wanted to touch base on and review again a little bit. Again, the renal pelvis is a structure which takes the filtrate. Filtrate's usually what it's called before it actually becomes urine. It's kind of a gray area. What's filtrate versus what's urine? They kind of end up being the same thing in the end. But the renal pelvis is what actually drains the kidney into the ureter, and from there the fluid is passed on to the bladder. We also mentioned the renal cortex versus renal medulla. The cortex is the outer part of the kidney, and we looked at some different animals like amphibians and reptiles, which only have nephrons to go into the cortex. They have no nephrons. If you look at a reptilian kidney, its nephrons never go down into this medulla. Never drop down that far. If you look at a bird, there might be a few that do that. If you look at your own kidney tissue, the cells, many of them are going to go not only from the cortex, but also dropping down here into the medulla. And again, that greater length allows a greater opportunity for filtration and also greater opportunity for reabsorption in the kidneys. So the main functions of the nephron, of the kidneys themselves, is to filter the blood. Okay, so we talked about different kinds of things which could be filtered out of the blood. Here we have an afferent arteriole. This would have branched off initially from the renal artery, carrying blood toward the glomerulus. Really, really high pressure here. This blood is forced against the walls of the glomerulus, and there's going to be some substances that come out of the blood. There's going to be water, there's going to be solutes, there may be amino acids, there may be glucose potentially, there could be ions, there could be H+, all kinds of things come out of the blood and go off into this first tubule. Now right away, some of those substances are going to get reabsorbed right back into the blood again. There might be hardly any time at all between when they come out of the blood versus when they go back into the blood. And that's kind of a necessity because we can only be so selective when it's time to filter things out of the blood. Oftentimes, if you're filtering one thing out of the blood, there will be other things that come along with it. So we want to make sure we get those other more valuable, wanted things back into the blood if at all possible. We have that reabsorption step like we talked about before. One of the things I didn't point out before on one of our pictures here was, if we go back and take a look at this picture here will work. We have this tubule dropping down, descending loop, ascending loop, and then we have all these capillaries, what we call paratubular capillaries. All these capillaries that are around the loop of Henle, around the loop of nephron, whatever you want to call it. These capillaries are available to reabsorb sodiums and calciums and water in particular. That's how things get back into the blood, because these capillaries here are circulatory tissue. So if there's things coming out of the tubules here, they're going to go right away back into circulation into the blood. So our second step then was reabsorption, and then our third step was secretion. So getting filtrate substances out of the blood and eventually into the form of filtrate, what would eventually become urine as well, in those kidneys and ureters and bladders and urethras, and then from there on out of the body. There could also be secretion of other things, right? So we talked about aspects of the kidney when it comes time for endocrine function. There are certain aspects of both the renal cortex and renal medulla that are involved in producing hormones or other substances that might be involved in allowing for communication within the body of a chemical nature. So kidneys are very, very important. If you look at each side, both right and left kidneys, each one is made up of about, give or take, 1,000,000 or so nephrons that are all there helping to filter the blood, reabsorb things back into the blood, and also allow for secretion to occur. The nephrons that don't go very deep into the medulla are called cortical nephrons. Cortical nephrons, the ones that are relatively short, they stay only in the cortex. So cortical refers to cortex. Those nephrons never really dip down too far into the medulla. So if you were to look at an amphibian or a reptile or most bird nephrons, what you're going to see is that most of them are of the cortical type. A juxtamedullary nephron, on the other hand, these are the ones that have the loop of Henle that drops way down into the medulla of the kidney and allows for a much more extensive filtration and reabsorption of kidney filtrate. We mentioned before also the afferent arteriole, which is a branch or branches off of the renal artery that bring blood into the glomerulus where it can initially be filtered. And then from there, the blood is filtered, like we said before, under high pressure against the capillary walls of the glomerulus to force fluid out. And whatever's in that fluid can then be handled properly. If it's desirable, if it's needed, we can get it back into circulation immediately. If it's excess, if it's waste, if it's something we need to dispose of, we can also dispose of it. So once it's filtered, then we need to figure out whether or not we want to keep it and then how we're going to make that happen. From there, any blood that didn't get filtered out of circulation by the kidney is going to go into what's called the efferent arteriole, and that's going to go into the paratubular capillaries. We talked about the vasa recta a little bit. The vasa recta is a structure in the juxtamedullary nephrons, which also helps with reabsorption of things. Bowman's capsule, again, is the section around the glomerulus, and from there we're down into the proximal convoluted tubule. And we talked about before, there's a descending loop of the proximal convoluted tubule. There's also an ascending loop of the tubule as well. It's going down versus going up from the cortex to the medulla, and then back up from the medulla into the cortex, respectively. All right, so from there we're going to go into the distal convoluted tubule, like I mentioned a second ago. And I should also mention, too, that there are different things that happen in each one of these steps. So the glomerulus we've talked about, it's the filtering part. Proximal tubules, they're involved with a lot of reabsorption. The descending and ascending loops, one of them is, one of them isn't really involved with much reabsorption. And then as we get into the distal convoluted tubule, that's kind of like the last final step where we start to see some reabsorption of water in particular for reasons that we'll talk about. Any water that's still in the tubule that maybe doesn't need to be in the tubule can still be reabsorbed even as late as the distal convoluted tubule. From there, anything left in the tubules is going to drain into the collecting duct, and from there, like we've mentioned a couple of times now, that material is on its way off to the ureter. Okay, so these collecting ducts from many, many nephrons are going to get bigger and bigger and bigger until eventually you actually have the renal pelvis, which drains the filtrate into the ureter. Now, if you look at the actual amount of blood that's filtered here, right, the amount of blood filtered in a day by your kidneys is around 2,000 liters. 2,000 liters, that's 1,000 2-liter pop bottles of blood passing through these kidneys each day. Now, suffice it to say, you don't have 2,000 liters of blood in your body, ever, right? You're nowhere near that amount of blood in your body. So clearly then, what your body is doing is it's filtering the same blood over and over and over and over. That 1 milliliter of blood that you have circulating right now in one part of your body is going to go through your kidneys over and over and over and over and over every day. And that's part of, making sure the blood is a relatively hospitable environment for cellular function, enzyme activity, and so forth all the time. If you look at how much water actually leaves the blood and goes into the filtrate, now it's not 2,000 liters. 2,000 liters is just what passes through the kidneys. Of all the stuff that gets filtered out, if you look at water, there's about 180 liters of water per day entering into the filtrate part of the nephron. So clearly, you're not going to be urinating 180 liters of water per day. If you did, you'd have time for nothing else, right? You'd have some severe issues on your hand if you try to urinate that much. So obviously then, if we're filtering out 180 liters of water per day, most of that has to be reabsorbed by the kidney tubules. You don't want that to become part of your filtrate because you're going to spend your entire day peeing and doing nothing else. The stuff that's not reabsorbed is going to be solutes, things that are in the glomerular filtrate that are maybe not as valuable, or things that are in excess, or things that might be causing an issue for the body, or could potentially cause an issue for the body. Those sorts of things, if they're not reabsorbed, can then go ahead and move on in the filtrate step by step by step until eventually they enter into the urine. From there, we're going to go into the proximal convoluted tubule. So that was a glomerulus. The proximal convoluted tubule, descending loop of Henle, and ascending loop of Henle, eventually collecting duct, are going to be involved with reabsorption, especially the proximal tubule, the descending loop, and the collecting duct. All right, so we're going to stop here, and I will pick up a third section of the urinary system here in just a moment.